Alright guys, welcome to 3.3. Today we're going to look at controlling cell division. Okay, so when it comes to the cell cycle, it needs to be fairly regulated, right? That means that there needs to be a way in which the cell can check and stop and pause the cell cycle at certain points to make sure that everything is going smoothly, and if it's not, we need to fix the problems, right? And so that's what these checkpoints are, okay? There are two ways in which the cell does this. It's internal control measures and then also external control measures, okay? Internal control measures encompass the cell cycle checkpoints themselves. Basically, every time DNA replication occurs, there can be these mutations that can occur and give rise in the DNA, and they're not great. You don't want mutations to occur when you're replicating the cell and the DNA, and so these checkpoints serve as a way of examining, pausing the cell cycle, and checking for damage in the cell and allowing those repairs to happen, right? We call it internal control because they happen in the cell and they are triggered by the cell itself. There are basically three checkpoints that occur throughout the cell cycle, okay? The first one occurs in the G1 phase, around about here, just before it's going to either enter into the S phase or the resting phase G0. The second one occurs at the end of the G2 phase, between G2 and the mitosis phase. And then the last one occurs inside the mitosis phase in metaphase. All right, let's have a look. So the G1 checkpoint occurs at the end of the G1 phase. It's known as the restriction point. At this stage of the cell cycle, it's going to pause and it's going to check for enough resources for the cell to divide, because it's about to replicate its DNA and it needs to make sure it has all of the resources it can to assemble those nucleotides, replicate the DNA, and then go into mitosis later on. So it's going to check for resources, it's going to make sure that the cell is actually large enough to do that, and it's going to run through the DNA and check for any sort of damage, okay? The second phase, the second checkpoint, the G2 checkpoint, occurs towards that transition between G2 and the metaphase, and it's going to check for DNA replication, making sure that now that the DNA has just replicated, there are no mistakes in it for damage, because once you go into mitosis and that nucleus divides, it's going to be separated into the two cells and you won't be able to reverse that particular process. It also checks that mitosis has all of the necessary proteins that it needs in order for it to then divide. The third one is the M checkpoint, right? This basically occurs towards the end of metaphase during mitosis, and it's known as the spindle checkpoint. At this stage, it's going to check that those chromosomes are aligned properly at the equator, and they're lying horizontal and it's all good, and that the spindle fibers are correctly attached to the chromatids. Because after this stage, and it divides, if the spindle fibers shorten, but the chromosomes are not attached properly, you could end up with a chromosome in the wrong place. So rather than each side having half, you might have a whole chromosome getting dragged to one side and then nothing being pulled to the other side, right? So those three checkpoints are important internal measures in which the cell can say, stop, go, we're all good, and so forth, okay? Now, external control measures are basically signals from outside of the cell that are either going to promote or inhibit that cell division process, right? If it inhibits the cell division, it could include things like contact inhibition, right? Contact inhibition is basically this phenomena where when cells get crowded and they go in contact with neighboring cells, it's actually going to slow down the cell cycle to keep it in interphase. That means that those cell-to-cell signals are going to trigger and inhibit cell division between these two cells so that they don't overcrowd. For example, you, the individual, might run and you trip and you get a little gash or you get a little cut in your hand and then your body starts to heal that cut. At some point, after it's kind of done healing, it's going to want to stop that cell replication process, cell division process, because if it keeps going, you're going to grow more than, the original amount that was there, that was damaged, right? And so contact inhibition plays a really important role in that. On the other hand, if it is promoting cell division and it wants your body to divide, there are these molecules, these protein molecules called mitogens, which are growth factors and hormones that are going to trigger and encourage the cell to undergo mitosis and cell division, right? So when cells die during that cut or that gash, the neighboring cells, up until that point, they're going to release mitogens, and it's going to stimulate tissue repair, right? So here's your cell, here's the plasma membrane, and this molecule, this mitogen is going to attach to a glycoprotein on the surface of your plasma membrane. That's going to initiate a signal transduction pathway and trigger other molecules inside the cell, like cyclines and CDKs, which I'll explain in the next slide. And they're going to encourage the cell to replicate its DNA and to undergo mitosis and to fill out the damaged location. And then once it fills out, you're going to get contact inhibition occurring in order to then stop that process so that you don't excessively repair a section that you don't need to repair, right? So those are the two ways in which external measures, things that are outside of the cell, are going to control that cell cycle within the cell. Now, in addition to that, there are other environmental conditions that need to be considered as well. Nutrients is really important. You can't divide cells if you don't give them the proteins that they need to divide, right? Temperature plays a really important role, and so does things like pH and acidity play really important roles to either promote or inhibit cell division. Now, there is a YouTube video showing you guys what contact inhibition kind of looks like, and I'm just going to let you guys watch that in your own time. I'm just going to move on to the next slide. So I mentioned these cyclins and these CDKs. Well, these molecules come under a branch of molecules called regulator molecules, right? And regulator molecules do as the name suggests, and they regulate the cell cycle. Cyclins are core cell cycle regulators. They are super important because they are going to drive the events of the cell cycle forward, right? So if it's in G1, it's going to move towards the S phase. If it's in the S phase, it's going to move towards the G2 or the M phase. And there are basically a group of cyclin for each of those associated phases, right? Here's the G1 cyclin, and you can see that it's going to occur in concentrations all throughout the cell cycle, with the most occurring at the G1 phase, progressing it through all the way towards the M phase. There's the G1S cyclin, then there's the S cyclin and the M cyclin. Now, I don't need you to know the specifics of what each one does, but having a clear understanding that these regulators will move and drive the process forward is really important. The second associated molecules are what we call cyclin-dependent kinases, or CDKs. The two molecules work together. Cyclins and CDKs work together. CDKs are a family of enzymes that work together with the cyclins to drive their cell cycle forward, right? How it works is that the cyclin is going to activate the CDK like a switch, right? And then that CDK cyclin combination, that complex, is going to target specific proteins within the cell that are going to activate the next phase of the cell cycle, right? So for example, right, if there is no cyclin, the CDK is inactive. That means the target proteins that it is trying to switch on are going to be switched off. And then that also means that, let's say, for example, if it is the S phase, that means the S phase is not going to start yet because these target molecules are not switched on because the CDK is inactive. However, let's say the G1S cyclin attaches itself to the CDK that now activates this particular enzyme. If it activates this particular enzyme, it's going to what we call phosphorylate the particular target molecules here, here, and here. For the sake of simplicity, we're going to call them target 1, 2, and 3. And then when these three things activate, they're all like switches. You switch all three of them on, and then the S phase begins, and the cell will start to replicate its DNA. Okay? And that replication of DNA molecules could include things like DNA polymerase, for example, like we looked at in the previous video. So the two work together, right? Now, there is another molecule that is very crucial, and that is the tumor protein 53 molecule, right? The tumor protein 53, also known as the guardian of the genome, or simply the P53, is a very crucial molecule that regulates the cell cycle. What happens is TP53 is going to activate when you get DNA damage, right? When you have DNA damage, it's going to pause the cell cycle, and it's going to activate repair enzymes that are going to fix that damaged DNA, right? So here is the checkpoint during the G1 checkpoint. let's say during that checkpoint, a couple of molecules identify that there is damage to the DNA here. That damage in the DNA is going to activate the P53 molecule, and the P53 molecule is going to activate what we call a CDK inhibitor. The CDK inhibitor binds to the CDK cycling complex and is going to, as the name suggests, inhibit it from working, which means it's going to deactivate the complex, and it's going to pause the cell cycle before it goes into the S phase. Okay, if there is too much damage, let's say it occurs here, here, and here, and here, and the cell deems that as a complete loss, it's going to actually program the cell to automatically die, and that process is called apoptosis. And so you kind of get these two pathways. DNA damage is going to occur. The P53 gene is going to activate. It's going to pause that cell cycle, and then it's going to release these CDK inhibitor enzymes that are going to then pause that cell cycle. Additional enzymes are activated to initiate DNA repair. And let's say it works. The enzymes fix the DNA. The CDK inhibitor gets switched off, releases from the CDK complex, and then the cell continues to move into the S phase, and you have DNA replication occurring. If it is a total loss, the P53 gene is going to activate other molecules that are going to induce the cell to basically kill itself, right? So that brings us to the process of apoptosis. Apoptosis is programmed cell death. It is a regulated form of a cell killing itself, but it serves a really vital function for organisms, right? These apoptotic enzymes called caspases are going to activate, and they're going to actually basically eat the cell from the inside out. The nucleus also collapses, and the cell is going to shrink. The plasma membrane kind of forms these little blebs and form little bubbles called apoptotic bodies, which are going to carry the remnants of the cell cytoplasm. And then the apoptotic bodies get eaten by these phagocytes or white blood cells in your immune system and digested, right? So basically, this is a cell that belonged to the body, and the body has triggered cell death, and then it breaks into these little blebs. There's actually some bacteria that you can see on those cells, and then these little blebs get eaten by the white blood cells in the body or the immune system. Here is a GIF of that process happening. So there's apoptosis there, that apoptosis there, and that's the white blood cell that was about to snatch it all up. Okay. Now, apoptosis is very, very kind of, a bit of a different mindset to have, right? Why on earth would a cell program itself to die? Well, it turns out that apoptosis is an important part of normal development, right? Examples of apoptosis serving normal development is actually found in, for example, webbing, right? Here is the development of embryo kind of mouse. And you can see this is the mouse paw, and you can kind of see the digits there. But, at the start, there's this webbing that occurs in between. Well, the body, as it develops the embryo, is going to trigger apoptosis in the regions between the digits. There, and there. Those regions are going to undergo apoptosis, and you're going to start to see the webbing formed between the digits, eventually giving the separation of the digits and the individual digits that the mouse is able to then use, right? Other examples will include things like nerve cell development, and then the neuron connections on the edge of each of those cells. Those little connections are these kind of frilly thingies. Well, the cell needs to kind of eliminate the in-between in order for the neuron connections to occur between the cells, and you get normal nerve development. There are basically two ways in which apoptosis occurs, right? It is either what we call intrinsic, or the mitochondrial pathway through the mitochondria, or it is extrinsic through the death receptor pathway, okay? In the mitochondrial pathway, when you have damage to DNA, a series of reactions are going to cause the mitochondria to stop functioning, right? When the mitochondria stops functioning, it means the cell cannot produce ATP, and it runs out of energy. And that running out of energy is going to release the same caspase enzyme that the cell uses to trigger apoptosis, and the cell dies. The second pathway to apoptosis is through the death receptor pathway, which is extrinsic. It's triggered because there is an external signal outside of the cell. For example, there's a lack of growth factors. The cell goes through hypoxia. It doesn't have enough oxygen. there's a viral infection or some sort of UV radiation that kills the cell. And those extrinsic pathways are going to trigger similar reactions, but they then release the same enzyme, the caspases, to kill the rest of the cell. And here is what a cell looks like when it goes through apoptosis. You can see there, that cell kind of pulls itself inwards, blebs. See those little apoptotic bodies that are kind of, bubbling around it. And then the cell kind of explodes, right? And that's how apoptosis works. Now, when it's not intentional and a cell dies by accident, we call that necrosis, right? Necro meaning death, and lysis, or the breaking up of the cells. So necrosis is the accidental death of cells. Usually some sort of rupture in the plasma membrane, allowing water and ions to enter into the cell, and it's going to swell and explode. Right? Here's an example of that. Here's a single-celled organism that's kind of just living its life, and then all of a sudden you start to notice that the plasma membrane ruptures. The cell then swells, and all of its contents just burst out into the nether, and it dies. Right? Variety of reasons why this would occur: physical damage to the cell, toxins that venomous and poisonous to the body, pathogens invading things that affect the body, lack of oxygen that then also trigger the death of the cells as well. So you can see that when the cell dies, if it dies via this kind of blebbing and then condensing and then fragmenting, that's apoptosis. But then when it swells, gets bigger, and then explodes, that is necrosis. Okay, on a slightly different note, when we look at cell division, it doesn't happen endlessly. There's a limit to when cell division occurs, right? Eventually, after a certain number of division, it starts to actually fail. In other words, your cells can't divide endlessly, but after a while, it actually slows down and it stops being able to divide and function as per normal, right? And scientists reckon that it's on the account of these particular sections called telomeres. Telomeres are these repetitive strips of DNA that sit at the very end of the chromosomes. And what they do is they act as a buffer against DNA loss during mitosis, right? So here's what it kind of looks like. So here's a chromosome, there's your centromere, and then there's your two chromatids, right? The black section in this diagram shows the telomeres. And what is generally happening is as the cells divide, right? Sections of the telomere is going to naturally shorten. It just can't help it. It's just going to shorten over time. And every time the cell divides, it gets a little bit shorter, and it gets a little bit shorter, and it gets a little bit shorter, until eventually it just disappears altogether. So eventually telomeres are going to disappear with age. And when the cell division process loses all of those telomeres, it's going to cut into the active genes that are responsible for the function of the cells. That means those genes are going to stop functioning because they're being chopped off every time the cell divides, and eventually the cell stops being able to function the way it should, right? So that is kind of the responsible, is what's responsible for the process of aging in people. So for example, your skin, when your skin ages, what you're likely to see over a long period of time, we're talking decades, is that the cells in between the skin, the dead skin cells on the surface and then the muscles below, the elastin and the collagen cells and all the stuff under there, they're going to, after a while, lose their telomere ends, and then the genes that keep the elasticity firm and rigid, they're going to slowly die off, and then eventually the skin starts to sag and you get to see the typical signs of aging. Okay? Now, on the note of cell division, we also want to take a bit of a look at cancer. Okay? Any sort of abnormal growth in tissues in the body are called neoplasms or tumors, right? We're familiar with the term tumors. Well, turns out tumors are not necessarily cancers yet, right? Tumors fall under three categories. They can be benign, where you have localized masses, but they just don't develop into cancer. You can have potentially malignant tumors, where they're localized and they probably will eventually turn into cancer. And then you have malignant tumors, which form masses that are going to invade other areas of the body and they transform into cancer, right? And so cancer is when you have an uncontrolled cell division, right? Where the cell is dividing and it keeps dividing and it doesn't stop for whatever reason, right? So here's how it starts. Here's a normal cell. Let's say, for example, your skin cells, and then let's say you sit in the sun, and the UV radiation from the sun is going to hit a section of your cells, at particularly the section that is not lathered with sunscreen, and it mutates the DNA. The UV radiation is going to mutate the DNA, and for whatever reason, it doesn't get repaired or checked in the checkpoints, and then it develops into malignant tumors, which become cancerous. And then this uncontrolled cell division keeps occurring, and it doesn't stop, right? It doesn't stop. So it starts to form this malignant tumor, and then eventually you're going to get sections of those cells break off and go into your transport vessels, like your blood or your lymph, and it's going to move around the body. So these cancerous these cancerous cells are going to move to a different part of the body, and it's going to cause that particular part of the body to also become cancerous, and the cell division there is going to become uncontrollable also, right? And when that happens, it's called metastasis, right? Where it starts to invade other parts of the body via the blood or the lymph vessels, okay? And that's how cancer typically spreads. It might start on the skin, but if it gets into your blood or lymph, it might travel to other parts of your body. Characteristics of cancer cells. Generally speaking, when cells become cancerous, they're going to divide much faster than your regular normal cells. They're not affected by normal signals that are controlling the cell cycle, like, for example, contact inhibition, right? So just because a cancerous cell kind of sidles up next to a regular cell, it's not going to receive those signals to say, hey, stop spreading because you're crowding up the place, and it just keeps going. Right? It's going to generally look different, and in general, the cells are going to be less specialized. They don't really function the normal things that they're supposed to do, but instead they just kind of keep dividing endlessly, right? The cancer cells also release these factors that are going to stimulate the development of their own blood supply, right? And that's because as the cells divide, they need blood, they need the nutrients from the blood to do that, and so they stimulate this development of their own blood supply so they can keep growing and dividing endlessly. In these cancer cells, the DNA mutates, which means that the DNA in cancer cells are going to be different and sometimes resistant to earlier successful treatments to those particular cells. And they can colonize new parts of the body and continue to grow unchecked, which is why early detection plays a really important role in slowing down or preventing cancers from spreading too much around the body. Right? And in the end, they can just kind of continue to divide endlessly. And through their own kind of mechanisms, they manage to avoid apoptosis. The cell is supposed to die, but it just doesn't die. Okay? Now, examples of cancer. This diagram shows a couple of cells that are cancerous. So you can see these are the normal ones, and then these are the cancerous ones there that are stained. This is leukemia, cancer of the blood. This is a melanoma. This is a mole on the surface of the skin, which has become cancerous and developing into a melanoma that is probably just under the skin. This is a brain tumor or a brain cancer up there. And then this is breast cancer. There is the lump of cells that are developing in the breast, leading to breast cancer. So when we think of cancer and the kind of pathways that cancer develops, right? Usually cancer develops as a result of genetic mutations, right? These are going to either increase cell division or it's going to suppress apoptosis, right? And if you think about it, it makes sense. It's going to keep dividing or stop dying, right? And it just keeps going. When you have apoptosis malfunctioning, right, and the cells doesn't die the way that it's supposed to, this causes that uncontrolled proliferation of cancer that you kind of see, right? So a few examples. So B-cell lymphoma, this one up here, right, occurs when you have excessive amounts of the anti-BCL protein, stops the mitochondrial pathway for apoptosis. So the cell is supposed to go through apoptosis through the mitochondrial pathway, but the excessive amounts of those proteins prevent that from occurring, and the B cells that sit in the lymph are supposed to die, but they don't, and then they become cancerous and survive. Other examples that can cause apoptosis malfunctioning is when the enzymes that apoptosis is supposed to release to kill the cell, they are mutated. So the genes that are responsible for producing these caspases are mutated, meaning these caspases enzymes are not working because the genes that make them are mutated, and therefore apoptosis doesn't occur because the enzyme's being released, but the enzymes don't work to kill the cell the way that it's supposed to, right? So those are examples of how apoptosis can malfunction. Some chemotherapy and radiotherapy kind of aim to induce this mitochondrial pathway so that in an artificial sense, apoptosis gets triggered and it eliminates the cancer, particularly before it starts to spread too far around the body. Now, on a slightly side note, excessive apoptosis is also really bad because, for example, it can be linked to neurodegenerative diseases like Alzheimer's, right? So here's a picture of a normal brain, and then here is the picture of brain and nerve cells undergoing too much apoptosis, and the nerve cells are dying when they're not really supposed to be dying, and you get things like memory loss and loss of function of motor skills, etc., etc. Okay, what causes cancer? Well, there are two factors. There are either genetic factors and then there are environmental factors. Genetic factors are mutations that occur in the genes that are responsible for cell division, right? And they cause cancer, right? And so when a gene is mutated and has the potential to cause cancer, we call those oncogenes, right? So onco is referring to the cancer, and then those are the genes, right? So when you go and see the doctor who is responsible for looking at cancer, we go and see an oncologist. And so oncogenes, looking at genes that have the potential to cause cancer. So types of genes in the body that can become, that are considered potentially as oncogenes include what we call proto-oncogenes. Now, proto-oncogenes are actually normal genes, right? They're involved in the regulation of cell division, right? And what they do is when they're there and they're triggered, they stimulate cell growth, or they're going to inhibit apoptosis and the body grows. These are normal. You're supposed to have normal proto-oncogenes that are functioning so that your body can grow and your cells can undergo replication. However, when you have a mutation of these genes, it can turn them into oncogenes and it becomes excessive and uncontrolled. There are another group of genes called tumor suppressor genes, and they, as the name suggests, they are going to suppress growth as opposed to promote it, like the proto-oncogenes do. These are also normal, right? So genes that code for proteins that are going to slow down the rate of cell division or the repair of DNA. So, for example, the gene that is responsible for producing the tumor protein 53, the guardian of the genome, that is a tumor suppressor gene, right? If you have a mutation in those genes, they also lead to cancer because these genes are supposed to activate and the proteins are meant to stop cell division. If they mutate and they don't work, it's not going to stop cell division when it's supposed to, and then you get excessive growth and cancer. A third category are actually inherited genes. Turns out there are certain forms of cancer that are specifically linked to certain inherited genes. For example, for breast cancer, there are two genes that are kind of identified as being linked. And they don't fully know exactly what these genes are responsible for, but we do know that they are linked to breast cancer. When you have these genes, in general, there is a good chance, or there is a likelihood that you will develop breast cancer. And we actually call them the breast cancer 1 and the breast cancer 2 genes, the BRCA1 and BRCA2. And here's just a diagram of breast cancer, and you can see there, that's the malignant tumor developing in the breast, and the presence of the BRCA1 and the BRCA2 gene, is linked to breast cancer. And so you can identify that in family lines. And so, if you see it found in mothers or fathers or whatever, there's a chance that maybe they might develop breast cancer. Now, the second category are environmental factors. Environmental factors are things external to the body that could potentially lead to damage to the DNA, right? Any agent that does this is called a mutagen, right? If the mutagen leads to cancer, then we also refer the mutagen as a carcinogen, right? So if something is carcinogenic, it means it is likely to cause cancer. Now, they can include chemicals. So for example, tobacco will cause cancer. Asbestos is especially nasty because asbestos are these fibrous materials with these tiny little hooks on them. And then when they disintegrate from the plasterboard or the asbestos board that you use for your insulation in houses, these little fibers have these little hooks. You breathe them in, they hook onto your cells on the inside of your lungs, and then your lungs actually grow over them. And because they are there, the growth of the tissues over that particular asbestos becomes mutated and cancerous, and that leads to lung cancer. Pollution, any sort of chemicals from the environment that gets into your lungs, gets into your body, into your blood, can also lead to cancer. Radiation is also considered as a mutagen, and UV radiation from being in the sun too much or X-rays also lead to cancer. And the reason for that is because these radiation, they have the potential to penetrate through your cells, hit your DNA, and then cause mutations in the DNA, which, if it's not fixed, leads to cancer. There are also biological examples. So, for example, certain viruses are actually likely to cause cancer, and we call those oncogenic viruses. Okay, so that's kind of how cancer really works. And that's it for this particular chapter. I'll see you guys in the next video for 3.4 when we look at stem cells. See ya.